Yeah, I was just we broadcasting live. They call this right now probably people that's like stuff to the guff from the you know what I'm saying? They, they got y'all as such. It's like y'all is like stuff to the guffs, stuff to the gut, not to the guffs. What's the guffs? To the gut stuff. I'm just chilling right now, and I'm just letting the warm man know that this gonna be out there for y'all. This the Church of United Skaters affiliation, and I don't give a shit what you talking about because you wrong as hell. And the word hell is he's ill, and you try to take the place of your creator, which was your mother, and you out your rabbit ass mind. And that's just the way it is, and it's gonna be like that. And I'm gonna keep saying it, and it's always gonna be here. And when I leave this and I come back, I'll be saying it again because this is my job. This was never your job. I've always been on the case. Like to the hot, to the pot, give you what you got, never want to stop. The truth will always be the truth. That's how that goes. I just want to say, أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين آمين صدق الله العظيم ويا أيها الذين آمنوا كونوا أنصار الله. So what I just said is what I just said. I said that all those fake people will be fake and all those real will be real. Allah has placed a seal on their eyes. They cannot see. They cannot hear. I can tell the truth all day and ain't nobody gonna hear. When the Most High wants you to hear, you gonna know. But for those who do know and that are afraid, it's time to get back to the truth. Now, it says, and the rib which the Lord Allah has taken from Adam made he woman and brought her to Adam. That don't make sense. See? Now, the only way a rib can make sense is the ribonucleic acid, which is for the DNA. Yeah. Hold on. I'll get the proper pronunciation. Because you're going to be like, what is he talking about? I got to be. Make me a car. proper pronunciation for you. Hold on. It's always something good. Okay. Look, it's so thick. One of those days, early in the morning, feeling so good, doing what you do. Okay, let's see if I can find him. Okay. Okay. This is like this is really going all up in it here. Okay. RNA. We're talking about the DNA. Let's see. So we got the RNA, but we're looking for the DNA right here. Ribno. RNA molecule. No, we're not talking about RNA. We're talking about DNA. Polynucleotides capable of directing their own synthesis. We're not talking about that. Chemical equilibrium. Not talking about that. We're talking about, okay. Let's just go from here. Okay, let's just go from here really quick. It says all living creatures are made of cells, small membrane bonded compartments filled with the concentrated aqueous solution of chemicals. The simplest form of life are solitary cells that propagate by dividing into two. Higher organisms such as ourselves are like cellular cities in which groups of cells perform specialized functions are linked by intricate systems of communication. Cells occupy a halfway point in the scale of biological complexities. We study them to learn, on the one hand, how they are made of molecules, and on the other hand, how they cooperate to make an organism as complex as a human being. Okay, so therefore, we're not dead, we're cells. Okay, the evolution of cell. I was talking about the DNA or the ribonic something. I'm gonna get that. Hold on. Let me see if I can go in the back and find it. CDFG, HJK, PRS, it takes time to study, you know. You gotta do your research. Never be embarrassed. Even though we're on the World Wide Web, it's okay. Alrighty, QRSTP. Okay, RARAS, rib protein, rib. Let's go to 1284. 1284, where are you? Let's see what 1284 says. Page 1284. Okay. It says DNA virus is the first group, but a general sense of describe applying to the most involvement in cancer. papillomaviruses, for example, are the cause of human warts and are also implicated in carcinomas of the uterine cervix. They are distinctly related to polyomavirus family that includes SB40. But we're not looking for that. It's just talking about the DNA tumors, stuff like that. We're actually just looking for the rib and how cells are created. Which I'm looking. Can't. Okay, hold on. Okay, good. Nope. It's not gonna give blood. See. Certain things I do not have knowledge of. You look for it and you find it. We're all gonna find out what it is. But to say that you were dead in your body and brought back to life, no. That was an idiot statement from an idiot man. Yeah, I got a lot of things on my mind right now. I'm trying to find, not the gene, excuse me. RNA and DNA. Close to it. A cell can change the expression of its genes in response to external signals. Genes expression can regulate it at many of the steps of pathway. Okay. Double helix structure of the DNA. Now we're getting to what we're talking about. Okay, DNA, regular proteins. How does a cell determine which of its thousands of genes to transcribe? As discussed in Chapter B, the transcription of each gene is controlled by a regulatory region of the DNA near the site where the transition begins. Some regulatory regions are simple and act as switches that are thrown by a single signal. Other regulatory regions are complex and act as tiny micro microprocessors responding to a variety of signals that they interpret and integrate to switch the neighboring gene on or off, whether complex or simple. These switching devices contain the two fun- fundamental types of components. One, short stretches of DNA of defined sequence, and two, gene regulatory proteins that recognize and bind to them. We begin our discussion of gene regulatory proteins by describing how these proteins are discovered. Gene regulatory proteins were discovered using bacterial genetics. Genetics analyses in bacteria carried out in the 1950s provided the first evidence of the existence of gene regulatory proteins which that turn specific set of genes on or off. Now we're talking about genes. Talk a little bit more. So, what we're talking about is the gene, the geometry of the DNA double helix depends upon the nucleotide sequence. Okay, short DNA sequences are fundamental components of genetic switches. So we have genetic switches. Okay. Gene regulatory proteins contain structural motifs that can read DNA sequences. The helix turn helix motif is one of the simplest and most common DNA binding motifs. Okay. What we looking for, what I had one is the rib. I still don't see rib. I still don't see rib in there. It's telling everything about it, but it is not telling me what I want to know. But it's okay. It should be like closer to the book. You know what I'm saying? To the book right there. Let's see, the amoeba. Okay. The eukaryotic cell depends on the mitochondria for their oxidative metabolism. Okay, they turn the mitochondria and say, well, let me go get this book and that book, you know, and that's all good. That's not what I'm looking for right now. Let's say, a lot of different things that you gotta study. This right here is the cell. I mean, there's a lot of things you gotta study if you want to know what you need to know. But here are some of the basic development programs attempt to consider evolution. Different things, different tadpoles. So how could we be dead? It's just that we're different creation now. And now we have supreme knowledge, so we way different than the animal kingdom. We're here to teach them because they're still evolving. That was the problem. All these slick ass people thought they was so slick. They ain't slick. How you doing? How you doing today? Evolution of a cell. Okay. It says all living creatures are made of cells, small membrane bonded compartments filled with concentrated aqueous solutions of chemicals. We read that earlier. Okay. It says this chapter, like the book as a whole, is concerned with the progression from molecules to multicellular organisms. It discussed the evolution of the cell, first as a living unit constructed from smaller parts, and then as a building block for larger structures. Through evolution, we introduce the cell components and activities that are to be treated in detail in broadly similar sequence in chapter that follows, beginning with the origin of the first cell on Earth. We considered how the properties of certain types of larger molecules allow hereditary information to be transmitted and expressed and permit evolution to occur. Enclosed in the membrane, those molecules provide the essentials of a self-replicating cell. Following this, we describe the major transition that occurred in the course of evolution from small bacterium-like cells to much larger and more complete cells, such as are found in the present-day plants and animals. Lastly, we suggest ways in which single-free Living cells may give rise to large multicellular organisms, becoming specialized and cooperating in the formation of such intricate organs as the brain. Clearly, there are dangers in introducing the cell through its evolution. The large gaps of our knowledge can be filled only by speculations. So this book is saying that they know, but they really don't know, but they speculate. They speculate. Speculation for the nation. Speculation. Things they say. I'm just looking at a book of speculation. They just say they really, 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 really don't know. But the red was the big red line inclusive something. I just can't find it right now. Yeah, let's see what they say here. It says bacteria are the simple organisms found in most natural environments. They are spheroidal or rod-shaped cells, commonly called micrometers in a linear dimension. They often possess a tough protective coat called a cell wall, beneath which is a plasma membrane enclosed in a single cytoplasmic compartment containing DNA, RNA, protein, and small molecules. In an electron microscope, the cell interior appears as a matrix of varying textures without any obvious organized internal structure. Bacteria are small and can replicate quickly, simply dividing into two by binary fusion or fission. When food is plentiful, survival of the fittest generally means survival of those that can divide the fastest. Upon optimal conditions, a single prokaryotic cell can divide every 20 minutes and thereby give rise to 5 billion cells, approximately equal to the present human population on Earth in less than 11 hours. The ability to divide quickly enables populations of bacteria to adapt rapidly to change in their environment. Under laboratory conditions, for example, a population of bacteria maintained in a large vat will evolve within a few weeks by spontaneous mutation and natural selection to utilize new types of sugar molecules as carbon sources, or spontaneous mutation. And that's what's happening with man. Man is a mutation from the woman, and he's trying to take her place, and we're not gonna let him. He think he's already did it, but that's not true. It's only in his mind, because he don't understand that his day, his 1,000 years is only one day, and we just got back. We left and came back, but they say it is written 6,600 years is a share. No, but it's a shame that they write some foolishness like that because time don't exist. We was here, we always was here. We went and left back in their mind. You see what I'm saying? But we was always here. Imagine if we wasn't here. So anyway, I'm gonna leave that to you right there. I'm gonna get right back with y'all. Let me right back again.